So I will present a bit exotic presentation because I will talk mainly about auditory fear conditioning and the way we have approached the probing of the neuronal circuits involved in fear conditioning. So just to start, I will turn the main question we are addressing in my lab. So in this scheme, you have neuronal circuits with different neuronal elements, excitatory, inhibitory neurons. Those circuits receive some sensory inputs. These inputs are processed within the network, and somehow a behavioral output is produced. So the main question we are addressing in my lab is how to relate the plasticity that develops in such circuits with behavioral learning. And as I said just before, we are using a really simple behavioral model that is the auditory fear conditioning. For those who are not familiar with this, it's a simple model in which animal is presented with a conditioned stimulus like a tone that is repeatedly associated with unconditioned stimulus like a food shock. And after this occurrence, the animal learns that the sound predicts the food shock. And on a test day, the next day, for instance, when you present back the tone, the animal will display a broad range of conditioned fear responses, including an immobilization reaction. That is a good indicator of the fear of the animal that is called the freezing response. And we quantify that to evaluate conditioned fear behavior. So this is just the conditioning, the pairing between the CS, the tone, and the shock QS. You get development of fear reaction with time. And interestingly, if you present repeatedly the same tone but this time without any adversive consequences in a different context than the conditioning context, you will induce a progressive decrease in conditioned fear responses, and this phenomenon has been labeled fear extinction. Next, if you introduce a delay between this extinction phase and retrieval phase, where you present back the sound, you will observe that not all the animal, but half of the animal will show a recovery of conditioned fear responses. And this phenomenon is quite important because it tells us that extinction is not an erasure of the original memory trace, but extinction is a new learning. And during retrieval, there is a competition between the fear memory trace and the extinction memory trace. And depending on the winner, the animal will show high fear recovery or low fear recovery. So which are the neuronal substrate of fear conditioning and extinction? So this is a coronal slice from a mouse brain where you see here the amygdala. And in fact, it has been shown that this region of the medial temporal lobe is really important for associative memories. And in the context of auditory fear conditioning, the information related to the sound and the food shock reach the amygdala via two main routes, directly from the sensory thalamus, or indirectly via the sensory cortex. Those information are processed within the lateral and basal amygdala, relayed to the central amygdala, that is the output station, that project to brainstem and hypothalamus structure that have been involved in the genesis of freezing responses. So if you make lesion or inactivation of this region, you completely abolish conditioned fear in mammals. And it has been shown also that the inactivation of one particular nucleus, the basal amygdala here, also blocked the extinction of conditioned fear responses. So it's known from this data that the basolateral amygdala is involved in both fear conditioning and extinction. It's involved for the acquisition, the storage, and the extinction of conditioned fear memories. So this is just a clinical point I wanted to make about the prevalence of anxiety disorders among pathological conditions, and you see that anxiety disorder are highly prevalent pathological conditions, and they are like the post-traumatic stress disorder, generalized anxiety, panic disorder, or OCD, and all these pathologies are associated with an hyperactivation of the amygdala. So the synopsis of my presentation today, I will try to cover 3 topics. The first one is related to the encoding of fear and extinction behavior in basal amygdala circuits. The second is related to more prefrontal circuits and the role of these prefrontal circuits in the regulation of fear expression after conditioning. And the last one, something that is not published, related to the role of prefrontal region in contextual fear generalization or discrimination. So I will focus mainly on the second part of this will be the longest part of my presentation. And this part would involve a lot of optogenetics. So I will discuss a lot about optogenetic here. For the first part, this work was done was published in 2008. And at this time, optogenetics were not really developed. So I will discuss some different tools we have been using to address which circuits are involved in fear acquisition and expression. Okay, and I start just with the encoding of fear and extinction behavior in basal amygdala circuits. So of course, just interrupt me whenever you want. So as I said earlier, the amygdala is involved in extinction learning, and this has been shown by pharmacological blockade. If, for instance, you inject an NMDA receptor antagonist or MAP kinase inhibitors directly into the basal amygdala, you block extinction learning. During my PhD, I was doing some CFOS experiment. CFOS was used as a marker of neuronal activity in this case, and we performed CFOS staining in the basal amygdala after extinction learning, and we observed that upon extinction, there was a strong increase in the expression of the CFOS in a particular region of the basal amygdala. So this prompted us to work in much more detail about the basal amygdala and which were the neural circuits involved in extinction learning. And to address this question, we use single units recordings in behaving animals. So in principle, the principle is quite simple. You put an electrode in the region of interest. In this case, it was the basal amygdala. You posit the electrode around some neurons, and you pick up the extracellular activity of those neurons that are action potentials. So on this representation here, you have there is a color code for the cells, and you have superimposed several action potentials that correspond to a single cell. So it's extracellular recordings, and you can see that based on the cell, the waveform shape is quite constant, and based on this constancy, you can establish cluster of action potential that correspond to single neurons, and you can follow the changes in the activity of these neurons during your behavioral experiment. So we have performed single units recording in the basal amygdala. These are the recording sites in this basal amygdala. And we have submitted the animals to auditory fear conditioning. So in this paradigm, we use 2 different tones. The blue one, the CS minus, that is the control sound that is not associated with the foot shark, and the condition tone in red, the CS plus, that is associated with the foot shark. So before conditioning, that is during the habituation session, if you measure the freezing responses, you see that the presentation of the tones does not induce any particular freezing behavior. But after conditioning, the animal discriminates between the CS and the CS minus and CS plus. And after extinction, the fear is gone. So you can acquire the fear and reduce the fear. So we have recorded 2 main kinds of neurons in the basal amygdala during this paradigm. The first kind of neurons, we call them the fear neurons. What I'm describing here is the changes in neuronal activity in average population. For the presentation of the CS minus in blue or the CS plus in red, and we are looking at the changes in activity before the tone presentation or after the tone presentation. So what you can see for these fear neurons is that you get an increased activity of these neurons when you play the CS plus after conditioning, but these neurons are silent after extinction learning. They are even inhibited. So this is the first category of neurons we recorded in the basal amygdala.
Can I have a question?
Yeah.
Do you think these neurons always respond to to the conditional cue, like if you would record from the same neuron, but from 24 hours later, would they respond?
So for this particular class of neuron, I can tell you that if you test the animal, well, the first thing is that you need to record the same neuron for several days. This is an issue. But if you manage to do that, for this particular class of neuron, we know that each time the animal displays fear responses, those neurons were activated.
So it's like a constant characteristic of that.
Yeah, that's exactly. But when the fear is extinguished, they do not respond anymore.
I have a related question.
Yeah.
What about allocation of new memories? So if you train the animals in a distinct the fear conditioning task with a different tone,
yeah,
do these neurons get reactivated in a new?
So we haven't performed this experiment. So this is just a guess. I would say that probably the neurons are activated also to different conditioning sessions, but we haven't tested it in particular. So they represent 15% of the neurons recorded in the amygdala. And the second category are labeled extinction neurons. In fact, they behave the opposite manner than the fear neurons. Those guys are activated only when the fear is low after extinction learning, but not when the fear is high. And they also represent 15% of the neurons. So this is just 30%. 50% of the neurons recorded were silent, and yeah.
Sorry. Would they respond to the context? Conditional context?
So in this particular experiment, we extinguished the animal in a different context than the conditional context. But this is not shown in this figure, but we have, after the end of extinction, we have tested the animal in the original conditioning context. And of course, when you replace the animal in the conditioning context, the fear recovers, and of course, you observe an activation of the fear neurons. Okay, so this is just a summary of what I was describing. In gray, you have the freezing behavior during the 2 sessions of extinction, and on top of it, you have the changes in neuronal activity for fear and extinction neurons. So when the fear is high, fear neurons are activated, but not when the fear is low. And when the fear is low, extinction neurons are activated, but not fear neurons. So they are inversely correlated during high and low fear state. And the second thing you can see is that when their activity crossover, you have to have a significant change in behavior. Okay, so to address the specificity of fear and extinction neurons, we design the following experiment. We perform a discriminative extinction. The question here was to know if the extinction and fear neurons belong to separate class of neuron or if they are the continuum of the same class of neuron. So what we have done, we have conditioned animals to 2 different tones, the CS1 and the CS2. Both were associated with food shock, but we only extinguish one. So we extinguished the first one, and what we did, we simply presented the second tone at the end of the extinction. So this is what is depicted here. You have the habituation, you have the fear response to the CS1 that we extinguished, and the fear response to the CS2 that was not extinguished. And of course, we recorded fear and extinction neurons with this paradigm. So what about extinction neurons? As expected, first, we concentrate on the CS1. This is the early extinction and the late extinction. So extinction neurons are activated when the fear is low during late extinction. In contrast, fear neurons are activated when the fear is high on the early extinction, but not when the fear is low. So what about what is going on when we presented the CS2 at the end of the extinction? This is what we observed, is that as soon as you switch from CS1 to CS2, you switch back the activity of the extinction neurons. They become silent. And in contrast, the fear neurons that were silent start to be activated. Okay, so the next question we asked at this time was whether or not fear neuron and extinction neurons were inhibitory neuron or excitatory production neurons, and how they were integrated into the neuronal circuitry of fear conditioning and extinction. And to address this question, we use the following strategy. Again, as I told you before, at this time in 2008, there was no, the optogenetics were not really developed. So how can you evaluate whether a neuron projects to a structure, that is, it is an excitatory neuron, or, and where exactly? So we designed the following experiment. Let's say that in red you have the fear neurons we have recorded, and those neurons, they can project to remote structure like the prefrontal cortex or the hippocampus, or they can receive inputs from these structures. So if you activate with electrical stimulation the fibers in the remote structure, you can drive two kinds of neuronal responses. The first one is called an antidromic response, where you induce a spike in the fibers that travels back to the soma. This is an antidromic spike. Or if you stimulate the fibers that project to these fear neurons, for instance, you will generate an orthodromic spike. And the difference between the two is that there is a synapse in between for the orthodromic spike. And this synapse induce a delay. After a delay, introduce a delay, and then the action potential that you generate will never occur or arrive at the same time. So there is a jitter due to this synapse. And this is clearly an example of an orthodromic response. So if you got this kind of response, you know that the neurons you are recording receive projection from the remote structure. And depending on the latency, you can evaluate more or less if it is a monosynaptic or a polysynaptic connection. With antidromic stimulation, an antidromically generated spike is much more interesting because there is no synapse, so it means that each time you will stimulate, the antidromically generated spike will always arrive at the same time. And another characteristic of antidromic spikes is that they follow high frequency stimulation. So this is an example of at 200 Hz, and you see that you get the first spike related to the first stimulation, the second to the second stimulation, and the third to the third one, with always the same latency in between. So when you get these particular responses, you are sure that the neurons you are recording from project to the structure you are stimulating. So this is a way to evaluate how a neuron can, whether or not a neuron projects to a different structure. So because we were in the basal amygdala, if you identify neurons that project to a remote structure, they are likely to be excitatory neurons. Based on using this strategy, this is the result we got. We stimulated the ventral hippocampus and the medial prefrontal cortex because both two structures are heavily connected with the basal amygdala. And for fear neurons, we observed that they receive strong input from the hippocampus and they project to the prefrontal cortex. In contrast, the extinction neurons are not connected with the hippocampus, but they project and receive input from the prefrontal cortex. So, something I didn't mention, of course, to realize this experiment, what you need to do is to first record the neurons during the behavioral experiment, and then we slightly anesthetized the animal, and we stimulated the prefrontal cortex or the hippocampus while recording from the neurons identified as fear and extinction neurons. Okay, so using this strategy, we were able to show that both fear and extinction neurons are excitatory neurons, and that they are differentially connected with the prefrontal cortex and the hippocampus. Okay, so the next thing we did in this experiment was to try to bring some causality again. There was no optogenetic available at this time. So it was not easy to target fear and extinction neurons. So what we have decided is to use a pharmacological approach. We inactivated the basal amygdala by iontophoretic injection of mucimol, that is a GABA A receptor agonist. And to do so, we use the following techniques. So we perform the iontophoretic injection with this device that we built. In fact, you have a glass pipette that was filled with mucimol, and around the tip of the pipette, we posit some electrodes to record the activity of the neurons. And the first experiment we did was to evaluate whether the mucimol injection was inhibiting neuronal activity. And that's exactly what you see. This is multi-unit activity before the injection, and as soon as you do the mucimol injection, you see that you reduce completely you don't see any more spikes. And this for 2 hours. So we have a temporal window of 2 hours to inhibit the neuronal activity within the basal amygdala. So this is the kind of injection we were able to do, really targeted injection, and we use for controlling the diffusion, we use the fluorescent mucimol, so you can precisely control the injection site. And this is the behavioral result we got at this time. So this is a simple conditioning experiment with 2 extinction sessions. We have the control group in blue, that is just the fluorophore injected animals. So if we concentrate on the control group first, after conditioning they display high fear responses, you can get extinction with time. And in comparison, if you inject the mucimol before the second extinction in the group in red, the mucimol injected animals, you see that you completely block extinction learning. And this was not an effect due to a lesion, because the next day in a drug-free situation, if you re-expose the animal to the sound, they extinguish the fear. Okay, so this demonstrates that the basal amygdala inactivation completely block extinction learning. So from those data, we have shown that there is subpopulation of neurons in the basal amygdala that specifically encode fear and extinction behavior. These data are interesting, but they also raise other questions. And the 2 questions we were interested in are the following one. What is the anatomical connectivity of fear and extinction neurons, the precise one, not only the mPFC, but which region of the prefrontal cortex, for instance, and is there any mean by which we could specifically manipulate those fear and extinction neurons during fear behavior? So to address this question, what you would like to do is to label the neurons, to inject the dye in the neuron, and here we have a problem because we use extracellular recording, so we cannot label the neurons. So how to turn around this question? We developed the following strategy. So what do we know about fear and extinction neurons? The first thing we know is that fear neurons project to the prefrontal cortex, and extinction neurons project and get input from the prefrontal cortex. So what we have done, we have used a retrograde tracer injected in the prefrontal cortex to back-label the cell bodies of the neurons projecting to the mPFC. And because different regions of the prefrontal cortex are known to differentially modulate fear behavior, in particular, there is the dorsal region of the prelimbic area that is known to, when you stimulate this region, you induce fear behavior. In contrast, the infralimbic region is known to be involved in extinction. If you activate those guys, this structure here, you induce low fear responses. So obviously, we injected the retrograde in the PL and the IL. And the second thing we know is that fear neurons are activated when the fear is high, after conditioning, and extinction neurons are activated when the fear is low, after extinction learning, or during extinction learning. So in complement to the retrograde tracer injection, we perform C4 immunostaining in injected animals. And here you have an example of a retrobead labeling of amygdala neuron in red here, coupled with C4 detection here. So these neurons has been activated during a particular behavioral task and is projected to the mPFC. So this is the strategy we have used to identify fear and extinction neurons. So these are the results for the C4 experiment. So we, of course, we quantify the co-labeling of the retrobid and the C4, and we did that after conditioning and after extinction. So first, when we evaluate the total population of prefrontal neurons, including PL and IL projected neurons, we observe that we get a strong C4 expression after conditioning and after extinction. But now, if we separate into PL and IL, we observe that the neurons from the amygdala that project to the PL were strongly activated after conditioning, but not after extinction. whereas the the neurons that project to the infralimbic area were strongly activated after extinction and to a lesser extent after fear conditioning. So these two populations, the PL projecting or the IL projecting, could correspond to fear and extinction neurons. That was the idea of this experiment. So we at this time, this paper was just published this year, beginning of the year. It has been a long story. This was a follow-up of the fear and extinction paper we published in 2008. But this time there was optogenetic available. So to perform those optogenetic manipulation of the PL and IL projecting neurons, we used the following strategy. So we injected a virus. We injected two viruses that bring the Cre recombinase to the soma of neurons in the BLA. So they are retrograde viruses. If you inject it in a targeted region, they will travel back to the soma and label the neurons. So we have used a combination of two. I will discuss that a bit later. We have used herpes simplex virus expressing Cre recombinase, or the CAF2 virus, that is a canine adeno-associated virus. Both are transported retrogradely, and here you can see, if you see that, you see the labeling of the neurons in the basal amygdala that project to the PL or to the IL. So this is an example of neurons we were able to drive using this strategy. So here you see each line correspond to, each dot correspond to action potential, and each line correspond to one trial. For of course, we have complemented the injection here in the basal amygdala with a Cre-dependent virus that was expressing both the channel rhodopsin and the allorhodopsin. So we could manipulate the neurons in two different ways in the same preparation. So, of course, in blue, you will activate the channel rhodopsin, in yellow, the allorhodopsin, and we identify neurons that we were able to drive neurons or to inhibit neuronal activity. And we have done also, we have coupled this injection with recordings of the neurons. So we have identified extinction neurons and fear neurons. And of course, not of course, but this was an output of the story, is that all the neurons that project to the IL, or I will put it in a different way, all the identified extinction neurons were neurons projecting to the IL. All fear neurons were neurons projecting to the prelimbic area. So, and then we, of course, we manipulated the IL and PL projecting neurons with optogenetics. And these are the behavioral results. It's just a summary, but in essence, following extinction, if we manipulated the IL and PL projecting neurons during extinction learning, we got an effect the next day. And I will concentrate here on the inhibition of the neuronal activity. If you inhibit the neuronal activity of extinction neuron, or IL projecting neurons, you induce an increase in fear responses the day after extinction. In contrast, if you inhibit the activity of fear neurons, you induce a decrease of conditioned fear responses after extinction learning. So these demonstrate that the optogenetic manipulation of prelimbic and infralimbic projecting basal amygdala neurons oppositely affect long-term extinction memories. Okay, so this was the first part about the fear and extinction neurons, and I will switch now to the prefrontal cortex. And I first, before doing that, put this scheme so to summarize what we have shown so far. So you have fear and extinction neurons in the basal amygdala. We know they project differentially to the prelimbic or the dorsal prefrontal cortex or the ventral infralimbic area. So fear neurons project to the prelimbic, extinction neurons to the infralimbic. And we know that the stimulation of these two areas produce different behavioral output. If you activate the PL, you induce high fear. If you activate the IL, you induce low fear. So we know that the prefrontal cortex is involved in the regulation of fear behavior, but what is not known is which are the neuronal elements, the particular neuronal elements, in particular for the prelimbic area, that are involved in the regulation of fear expression. So this is just what I was saying. So if you perform inactivation of the mPFC, you block fear expression. If you perform electrical stimulation of the prelimbic area, you induce fear behavior. And from the group of great work in Puerto Rico, they have shown that you have neurons which neuronal activity correlate with fear expression in the prelimbic area. So this is the background. And as I said, the particular neuronal elements involved in the prelimbic area are completely unknown. So that was the question we were addressing in the study I will describe now. So like in any other cortices, the prefrontal cortex contains both excitatory neurons, in red here, and a myriad of inhibitory interneurons that represent 20% of the cell population. And I will focus particularly on the inhibitory neuron in blue that are called the basket cell and the chandelier cell because they target the soma of the excitatory neuron and the axon hillock. And with these properties, they can precisely control the output activity of the principal neurons. So they are really interesting candidates because they can block or allows the neurons to fire. So we know from anatomical studies that the pyramidal excitatory neuron in the prefrontal cortex, they project to both the basolateral amygdala and the periapical gray, and to many other structures, but I'm just mentioning the ones that are involved in fear behavior, the basal amygdala and the periapical gray. As I said, there is different population of interneurons that regulate the activity of principal neuron at different levels, at the soma, at the dendritic level as well. And the basket and the chandelier cell that control the cell body are known to express parvalbumin, so they are PV expressing interneurons. So we, in this study, we asked the question of what was the role of prefrontal inhibitory interneurons during conditioned fear behavior, and in particular of the role of the PV expressing interneurons. So again, we use a single unit recording as a start. This time it was not implanted in the amygdala but the dorsal prefrontal cortex, and again we were able to follow the activity of single neurons during the session. The behavior was the same. We used the classical fear conditioning with an habituation session where we presented the control sound, the CS minus or the CS plus. We conditioned the animal to the CS plus only, and we have 2 days of extinction, extinction 1, extinction 2. After extinction 2, you get a significant reduction of conditioned fear responses. And if you test the animal in the extinction context, 7 days later, you get a recovery of conditioned fear responses. So this is the classical behavior, and we recorded the activity of interneurons and pyramidal neurons during each of the behavioral sessions. So this is where we have done the recordings in the dorsal prefrontal cortex, the cingulate area and the prelimbic area. We were able to record around 700 neurons in these structures, and among those neurons, around 67% were tone-reactive neurons. The other one were just not reactive. So the first thing we did, we tried to separate the neuron into putative excitatory neurons or putative inhibitory interneurons using electrophysiological criterion. The first criterion was the firing frequency of the neuron. The second one was the spike half width, and the third one was the curve, the area under the curve that is supposed to correspond to the after hyperpolarization from spikes recorded intracellularly. So using this strategy, this is a completely unbiased methodology. We are not selecting the neurons that belong to each class. It's an automated process. And you can clearly see 2 clusters of neurons, in blue the one that corresponds to the putative excitatory neuron, and in red the one that corresponds to the putative inhibitory interneurons. And characteristic of the putative inhibitory interneuron is that the half spike width is shorter than for putative excitatory neuron, And we observe also really fast spiking neurons in the putative inhibitory cluster, the putative inhibitory interneuron cluster here. So using this strategy, among the 493 tone-reactive neurons, 29% were classified as putative inhibitory interneurons, and the remaining were classified as excitatory neurons. And this roughly corresponds to what has been described in the literature about the proportion of excitatory and inhibitory neurons. So for for this particular study, as I said, we focus on the inhibitory interneurons here. And we wanted to know what was the firing pattern of the neurons during behavior. So for this, we use an unbiased methodology called the principal component analysis to identify the firing pattern of the neuron in response to the tone presentation. This is for interneurons. You have the tone onset here, and this is the two main patterns we observe for the inhibitory interneurons. Patterns we call the type 1 and type 2. Type 1, the neurons are excited following the tone onset, and type 2, they are strongly inhibited. This is an example of such neuron, the excited one, type 1, or the strongly inhibited one, the type 2. And this is the population of neurons we recorded. So we got 68 type 1 interneurons, putative interneurons, and 15 putative type 2 interneurons. So as you can see on this graph, those two types of interneurons display antagonistic firing patterns. That's something I will come back to that in a moment. Okay, so what are the characteristics of these type 1 and type 2 interneurons? First, we wanted to know whether they were segregated within the cingulate or the prelimbic area, but we found that they were present everywhere in the cingulate and the PL. So there was no particular layers where those neurons were present or not, but they were everywhere in the cingulate and prelimbic area. We look at the firing rate to discriminate between type 1 and type 2 interneuron, and we observe that type 2 interneurons display fast firing patterns in comparison to type 1 interneurons. So this distinguishes what we call the regular versus the fast spiking interneurons. And I remind you that I mentioned that in the past that we observed within the the two clusters of excitatory and inhibitory neurons, we observed some neurons displaying a high firing rate, and basically this is those guys here. So again, we have the type 1 and the type 2 interneurons. And we went a bit further into the analysis. We look at whether or not they were phase locked to some oscillation, because this phase locking is characteristic of some interneurons. So for type 1, this is a theta oscillation, and on top of it you have the firing activity of the neuron. What we observe for type 1 interneuron is that they were not phase locked to the oscillation, to the theta oscillation. In contrast, we observe that the type 2 interneurons were phase locked to the peak of the oscillation. So this is a difference between type 1 and type 2. Type 2 interneurons are phase locked to theta oscillation. And this is more visible on this spectrogram, where you clearly see the locking of the neuron to a particular band that is in the 8 Hz range, that is the theta oscillation. And from the literature, this has been described in the cortex as being as corresponding to parvalbumin expressing interneurons, and these to non-parvalbumin expressing interneurons. So using just those criteria, we have a clue that type 2 might be PV expressing interneurons, but not type 1. And of course, to identify this in a more precise manner, we switch to an optogenetic strategy this time. So we use mice that express the Cre recombinase under the control of a parvalbumin promoter, the Cre-PD mice, in which we perform a conditional expression of archaeorhodopsin. So we injected the Cre-dependent virus in the prefrontal cortex to label with archaeorhodopsin only the PV interneurons. So I won't go into the detail of optogenetics because Alex provided a clear description this morning about how it works. I will just discuss what we observe. here. So this is an example of a type 2 interneuron that was identified based on those criteria. And this is the control situation. We don't have light. So each line, again, is a trial where we have the neuronal activity. And you see that under the control condition, there is nothing, of course. But as soon as we switch on the light, that is, we inhibited PV interneurons were able to identify this guy as a PV interneuron. So so this is one example, but we did that for a certain amount of neurons. We have an N of 5 here where we were able to identify the type 2 interneurons as being PV expressing interneurons, and none of the type 1 interneurons was showing any inhibition, indicating that type 1 are not PV expressing interneurons. So from this optogenetic experiment, we were able to say that the type 1 and type 2 interneurons we recorded from the mPFC where the type 2 were PV expressing interneurons. So when I'm saying that type 2 are PV expressing interneurons, I'm not saying that all PV are type 2 interneurons. I will explain this in a detail. So we know that the type 1 interneurons are not PV expressing interneurons. And among the PV interneurons, we were able to identify some were type 2 interneurons, but others were not identified. So what I'm saying here is that, what I've done here? Sorry. What I'm saying here is that the type 2 interneurons are a particular class of PV interneurons. You have several class of PV interneurons. Only the type 2 class of interneurons were inhibited during tone presentation. Those guys that were also PV interneurons were not inhibited during tone presentation. So type 2 are a particular class of, a functional class of PV expressing interneurons. Okay, this is just some control we provide for some anatomical controls. What we have done here, we have injected the viruses expressing the RHT in PV3 mice, and we wanted to control that all the cells labeled were PV neurons. So we co-label PV, and you have the overlap here. And in fact, we observed that 99.9% of the GFP positive neuron, that is the neuron expressing RHT, were also PV expressing interneurons. And we have done some slice physiology experiment to evaluate whether the expression of archaeorhodopsin in those PV neurons will change their firing pattern. So maybe we were able to record currents in activated or inhibited neurons using channel rhodopsin or archaeorhodopsin. But this is maybe the more critical experiment we have done. We have recorded from GFP neurons, so PV expressing just GFP, PV expressing channel rhodopsin, or PV expressing RHT receptor. And we have performed current injection and measure the firing frequency of those neurons. And there was no difference between those three categories of PV neurons in following the current injection. So they behave the same way than the neurons that were not infected with archaeorhodopsin. That was an important control for us. Okay, I mentioned earlier that type 1 and type 2 display antagonistic firing patterns. So this suggests that maybe type 1 could inhibit type 2 interneurons. And how did we address this question? We used the following strategy. The first thing we have done, we compare the activation and the inhibition latency of those neurons. So in type 1 you have the excitation, in type 2 the inhibition, and you see that there is a kind of perfect match between the latency of those two neurons, indicating that type 1 could inhibit type 2 interneurons, but we were not, this was not enough for us. What we wanted to do is to record in the same animal from type 1 and type 2 interneurons, and on those guys, on those animals, we have evaluated the firing latency of type 1 and type 2 interneurons. And we observed that type 1 interneurons fires always before type 2 interneurons, suggesting that they can inhibit type 2 interneurons. Okay, so this was just the characteristic. We know now that type 1 are non-PV expressing interneurons, type 2 are PV expressing interneurons. And the question was, what was their behavior during presentation of the CS plus or the CS minus? So this is, again, the two examples, type 1 and type 2. And this is the response to the tone presentation. In blue, the CS minus, the control tone, or in red, the CS plus. So you see that for the type 1, those guys were strongly activated to the CS plus in comparison to the CS minus. And for the type, they were strongly inhibited to the CS plus in comparison to the CS minus. This is the quantification of it. We have on the same graph in gray the freezing behavior during the CS minus and CS plus, so low fear, high fear. And in top of it, you have the changes in neuronal activity during the first 200 milliseconds for type 1 and type 2 interneurons. So for the type 1, you see there is an almost perfect match between the behavior and the change in neuronal activity. When the fear was low, the neurons were not activated. When the fear was high, they were activated. And the type 2 were inversely correlated with freezing behavior. That is, when the fear was low, they were not inhibited, but when the fear was high, the type 2 was strongly inhibited. So this is just the conclusion. Yeah.
How would you comment that you have quite a robust reaction to CS minus?
So as you have seen, maybe in the if I go back just this, to that. Okay, this is the behavior. In fact, you see that you have some residual freezing to the CS minus. They are not at 0; they are at around 20%. So some animals may display higher freezing responses to the CS minus. So it's why you see this this response to the CS minus in this example here. So I'm not saying that the animal do not freeze at all during CS minus. They could freeze a bit, right? So it's why you get this slight increase. But the point is that we know we have a significant difference in freezing behavior between CS minus and CS plus, and this is evident also in terms of neuronal activity.
Could you alternatively propose this attention?
So we thought about this question. That's a really good point. Something you can see here, especially for the type 2 interneurons. Can you see it here? Okay, so you see that in the first, in the first 10 milliseconds, the blue here is really, there is a strong inhibition to the CS minus, right? And in fact, if you consider the 10 milliseconds after the tone onset, there is no significant difference between the inhibition induced by the CS minus and the CS plus. And we think that this is reflecting attentional processes. What is occurring within the 10 milliseconds. But what is occurring later on is not related to attention because you have a differential effect depending if you present the CS minus or the CS plus. But definitely we think there is some attentional processes going on here, especially in the few 15 milliseconds after the tone onset. So the question here is, okay, we have the firing pattern of the neurons, and the question we wanted to address is whether this was a reflection of the freezing behavior, or if this was causing the freezing behavior. But that was the question. Does tone-evoked neuronal activity in type 2 interneurons mediate freezing behavior or the reverse? And the approach we used to address this question was the following one. We are not using pure tones in our protocol. We are using a series of 27 pips, so the tone goes like pip, 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 pip during 30 seconds. Of course, during this tone presentation, we can extract the freezing period when the animal is immobile. And of course, we can extract the pips that are inside the freezing period, outside the freezing period, or just before the freezing period. The idea here is being that if the changes in neuronal activity predict the changes in fear behavior, this should be obvious when selecting the first beep before the freezing episode. So using this strategy, if you consider the pips inside the freezing period, you can find back what we observed for the type 2, that is an inhibition. If you take the pips outside of the freezing period, there is no inhibition. But interestingly, if you take the first peak before the freezing period, you can already find the inhibition, suggesting that the changes in neuronal activity in type 2 interneuron precede and predict cognition fear behavior. So this is just the quantification, and this is the conclusion from that. So this is the first approach we use to this question. The second approach was an optogenetic approach. So now what I will discuss is the optogenetic manipulation of PV interneurons. And again, when I'm saying optogenetic manipulation, I want to say that we manipulated the activity of the PV interneurons within the 200 milliseconds to match perfectly their firing pattern. We are not inhibiting or activating the PV continuously. It's really precisely to match their firing pattern. So what we have done first is the manipulation of the inhibition of PV interneurons using arterial. This is the behavioral protocol we have used. We have a pre-conditioning session, a fear conditioning session, 24 hours later a post-conditioning session, and again 24 hours later an extinction session. The first manipulation we have done is during the pre-conditioning session. Without any conditioning, if you inhibit the PV interneurons, this is the effect we observe. We induce fear behavior without any conditioning. The second manipulation was we conditioned animals and we performed an extinction, so the fear was gone. And at the end of the extinction, when the fear was gone, we inhibited the PV interneurons, and the result of that was an increase in freezing behavior in comparison to control animals that were infected only with GFP. We did the opposite manipulation. We activated the PV interneurons. So this time we conditioned animal. We had a post-fear conditioning test during which we manipulated the neuron, and a 24-hour test later on. So this is the result following conditioning. The animal freeze to 80%. And if you activate the PV, the one we think are type 2 interneurons, you reduce condition fear responses significantly. And the next day, when we test the animal, there was no more difference between the presentation of the CS, indicating that this effect was really transient. So those two experiments demonstrate that the inhibition of PV expressing interneurons is both necessary and sufficient to drive fear expression. So this is the interpretation we have. There could be a second interpretation that could be more problematic. In fact, when we inhibit the PV interneurons, we induce this freezing behavior, and we think this is freezing behavior. But someone could say, okay, this is not freezing behavior, you're just altering the mobility of the animal. So this is just a motor reaction, it has nothing to do with fear conditioning. So that was important to control. And to control for that, we used a different behavioral task, that is a place avoidance task, where we manipulated the PV interneurons, so we inhibited the PV interneurons. So the animals were pre-exposed on day 1 to a two-chamber task, maze, and they could freely explore the compartment 1 or 2, and following the first day, we selected the compartment where they spend most of their time. And this was labeled as the preferred compartment. So the next day, what we have done, each time the mouse was entering the preferred compartment, we inhibited the PV interneurons, and we observed the behavioral effect. And what we observed is that from day 1 to day 2, when you inhibit the PV interneurons during day 2, the animal avoid the compartment in which the PV was stimulated, indicating that the PV inhibition per se does not induce motor alteration, because in this case, the animal was avoiding the compartment. So this rule out the possibility I was mentioning before, that this effect could be due just to motor impairment. So what is the functional relevance of this prefrontal inhibitory circuit? So this is just the model we had in mind at this time. You have two conditions, the CS minus condition and the CS plus condition. When you play the CS minus, this is associated with a low fear state. When you play the CS plus, this is associated with a high fear state, and this is the three types of neurons we have recorded in the mPFC. Type 1 and type 2, and also the putative excitatory neurons. So during the CS minus condition, when the fear is low, the putative, the type 1 are not activated, so the type 2 are not inhibited, and there is a tonic inhibition from type 2 to the projection neurons. When the fear is high, when it is induced by the CS+, we observe that we have an increased activation of type 1 that inhibit type 2 interneurons, and this inhibition induce because those guys are inhibited, they cannot inhibit anymore the projection neurons, and this phenomenon is called a disinhibition process. You remove an inhibition onto some neuronal population to make them activated. So this is the model, and this is the actual result. When we look at the projection neurons recorded, we observe that during the CS+ presentation, the projection neurons, the excitatory neuron, were strongly activated in response to the CS+ and much less to the CS-. So consistent with the disinhibitory process we have in mind. So we use also Optogenetic to demonstrate that the PV interneurons directly contact the principal neurons. So this is an example of a neuron here. During CS+ presentation, this is an excitatory neuron. You see that when you present the CS+, you get an activation in this neuron. If you associate the tone presentation with the activation of PV interneurons, you block this activation. And this is the total population. So this clearly demonstrates that PV interneurons can directly control the activity of principal neurons in the prefrontal cortex. We did the opposite strategy to evaluate a bit more the disinhibitory process. We use archaeorhodopsin stimulation. Again, this is an example. This is a pyramidal neuron during no light condition, nothing, and a pyramidal neuron during light inhibition of PV interneurons. If you inhibit the PV interneurons, you disinhibit the principal neuron. And this is the entire population. And this is with a really sharp latency, less than 2 milliseconds, indicating that this effect is monosynaptic. So the conclusion from this is that inhibition of PV expressing interneurons disinhibits prefrontal principal neurons during fear expression. Okay, so maybe you have observed, I was describing this principal neuron pattern. So you see that the neuron is activated at the tone onset, but there is like a rebound of activity just after, here and here. It's maybe more evident on this example, where we compare the activation of the putative excitatory neuron to the CS- and the CS+. Again, CS- is associated with low fear, CS+ is associated with high fear. So what you see clearly the disinhibition here on the CS+ onset, but also that the neuron is oscillating in this portion of the graph. So this is more visible on this example where we recorded 9 neurons in a single animal during CS- and CS+ presentation. During CS-, when the fear was low, the neurons are not doing a lot of things, but when you present the CS+, you see that those neurons are activated and they start to oscillate. So because the oscillation of principal neurons has been linked somehow to the local field potential, also has been linked to some oscillation in certain frequencies. And because here you can see that the oscillation occurs every 10 milliseconds, every 100 milliseconds, consistent with the theta modulation. So we evaluated the change in the local field potential using the same recordings in the animal, and we compare first the CS- and the CS+. So this is the local field potential filtered in the 8 to 12 Hertz range, in the theta range, high theta. During CS- presentation, you don't see anything occurring in the LFP, but during CS+ presentation, you see that you have a transient increase in the amplitude of the oscillation just after the tone onset. This is for a single pip here, but if you look at the 27 pips we have been using for our tone presentation, this is just the superimposition of the LFP for the 27 pips. You can see that there is nothing occurring in the CS- condition when the fear was low, But when the fear is high, we observe that there was a resetting of the oscillation. That is that you have a transient increase in the amplitude that always occur at the same time. And of course, this was linked to the firing of the neuron just in the peak of the oscillation. This is the methodology we have used to quantify the resetting of the oscillation. We have calculated the variance of occurrence of the first peak or the second peak. In case of low variance, this indicates that the peak always occur at the same time. So we have a low variance for the CS plus condition and a high variance for the CS minus. And this is the quantification of the resetting to the CS plus. You have a low variance for the CS plus. So this is just to just indicate that the tone presentation during high fear state induces a phase resetting of theta oscillation. So you probably know that theta oscillation have been strongly linked to the hippocampus. So the obvious question we had here, whether or not this theta oscillation recorded in the prefrontal cortex was coming from the hippocampus. So in other words, is it locally generated or is it generated in the hippocampus? And to address this question, we use again a targeted inactivation of the region involved in the genesis of theta oscillation, that is the medial septum. We perform, this is the cannula track, and we injected the fluorescent mucimol in the medial septum, and we performed the following experiment with fear-conditioned animals. We tested the animal before mucimol injection, during mucimol injection, or after mucimol injection. And these are the behavioral results. Following conditioning, all the animals were conditioned independently of the group, so the mucimol had no effect on the behavior. However, when you look at the local field potential in the hippocampus, we observe a strong reduction of the amplitude of the oscillation in the hippocampus. So basically what I'm saying is that when we inactivated the medial septum, we reduced the theta oscillation. Okay, so this was the effect. And now we evaluated the resetting of theta oscillation in the prefrontal cortex to see if this manipulation will alter the resetting of the oscillation. And we didn't observe any effect of the manipulation of the medial septum. So the resetting of the oscillation was still there while during the reduction of the theta oscillation in the hippocampus, suggesting that this theta resetting, that the theta observed in the prefrontal cortex does not come from the hippocampus, but could be rather locally generated. And we made the following observation. We manipulated the PV in 10 neurons, and we look at the effect of this manipulation to the local field potential. And this was really an interesting observation. This is the 27 local field potential after in the control condition. There is no light. And when we inhibited the PV in 10 neurons, we observed that we got the resetting of the oscillation. So here we are manipulating the PV in the prefrontal cortex. So we induce the resetting of the oscillation. And really interestingly, if you increase the length of the light pulse, you increase the number of oscillations. So this clearly indicates, and this is the control condition where we present a CS plus that is associated with a high freezing level and associated with the resetting of the oscillation. And if we activate the PV, at the same time we block the resetting. So this clearly indicates that this resetting of theta oscillation is due to the inhibition of PV in 10 neurons in the prefrontal cortex. So this is the conclusion. So this is almost the last part of this section of my presentation. Maybe I just skip this and I concentrate on that. We wanted to address what was the functional consequence of theta resetting at the level of medial prefrontal cortex neuron, in particular principal neurons. And in this representation, what you see is the resetting of the theta oscillation when the fear was high and no resetting when the fear was low. And we look at the phase locking of the neurons, the 300 pyramidal neurons, for 1 theta cycle before the tone onset and 3 theta cycle after the tone onset. And what you clearly see is that when you have a resetting of the oscillation, you see that the neurons start to fire in the peak of the oscillation, right? So this resetting of theta oscillation induces a synchronization of prefrontal output neurons. So maybe I just briefly mention something about. So the next question was, okay, those pyramidal neurons get synchronized during high fear state due to the resetting of the oscillation that was mediated by PV interneuron inhibition. The question is, where do these neurons project to regulate fear responses? And again, we used the antidromic stimulation strategy, and we stimulated the fibers in the BLA or the PAG, periodical gray, because we know that the prefrontal cortex projects to both regions. And we got antidromic responses, and what we observe is that all the principal neurons that were activated during the tone presentation were projecting to the basolateral amygdala. So if I put all together the result with this scheme, you have the type 1 interneuron in red that contact the PV interneurons in pink that control the activity of principal neurons that project to the amygdala. This is the scheme we think we have described, which is the neuronal circuit we have described, and this is the physiology of those neurons. Type 1 are excited during the CS plus associated with high fear state. Type 2 are inhibited, and therefore this inhibition disinhibit principal neuron that project to the BLA. This activation of principal neurons is locked to the oscillation, and we know that the PV interneurons manipulation induce the resetting of the oscillation that synchronizes the activity of BLA producing neurons. So these are just the conclusion of this study, that the identification of two populations of prefrontal interneurons differentially modulate fear behavior. The inhibition of type 2 PV interneurons is necessary and sufficient to drive fear expression. We have described a disinhibitory mechanism that facilitates principal output activity during high fear states. Those PV interneurons also mediate local theta phase resetting during freezing behavior that promote the synchronization of principal neuron output activity. And we think that we have two co-occurring mechanisms. The first one is the PV-mediated disinhibition and the theta phase locking of the principal neurons that allow the synchronization of those neurons to drive fear expression. That's the main conclusion of this study. So as you have seen, we have the strategy we have used in this experiment was to first try to identify the firing pattern of the neurons. This was the first thing, and then to manipulate optogenetically those neurons specifically during the phase where they were activated or inhibited. Okay, so I don't know what time is it now. So the last section I wanted to cover was also to the role of prefrontal cortex neuron, but this time in a bit different behavioral paradigm during contextual fear behavior. There is much less data available about the role of prefrontal cortex during contextual fear generalization. This is an important topic because fear generalization is a core feature of anxiety disorders, such as post-traumatic stress disorder, so it's important to understand which are the neuronal circuits involved in this phenomenon. So what do we know about contextual fear behavior, and in particular contextual fear generalization? So there is, here in this circuit, you have the prelimbic area of the prefrontal cortex, the hippocampus, the BLA, the periodical gray, and one nucleus in the thalamus called the Reinians nucleus. So first of all, if you make lesion of the prelimbic area here, you induce fear generalization. There was also a paper published by the Sudoff lab in 2013. They demonstrated that if you block the pathway from the prelimbic area to the thalamus, to the Reinians nucleus in the thalamus, you also promote fear generalization. And in fact, they came with the idea that the integrity of this pathway from the prelimbic area to the radiance back to the hippocampus and back again to the prelimbic area was important to provide a fine representation of the context to allow the animal to discriminate or general how to discriminate the different contexts. What is unknown is at the level of the prelimbic area, which are the circuits and elements that allow the expression of fear, particularly the fear generalization and fear discrimination. And this is this question we addressed in the following study. This is completely unpublished. So to study contextual fear conditioning, and particularly contextual fear generalization, what people are doing, usually they condition the animal in a given context. So contextual fear conditioning, you just may associate the context with food shock without sound presentation. So people condition the animal in one context, they extract the animal and they put the animal in a different context for testing. So when you are doing that, you cannot control when precisely the animal detects the behavioral change, the contextual changes. If you want to do single units recording, what you like to do is what you like to see is when, in terms of neuronal activity, the animal understands there is a change in the context. So we design a particular behavioral task to evaluate fear generalization and discrimination in the rodents. Basically, we leave the animal in the same context, but we change the contextual feature in order to create different contexts. So we condition the animal in one context, the A. This context is composed of light, some line odor, and sound. This is just the habituation. They are habituated to that for 10 minutes. And then on the conditioning day, they receive a food shock in this context. And the day after, we start the experiment by exposing the animal in the same conditioning context. And progressively, we remove some elements of the context while the animals stay in the same environment. So in the what we call the context B, we remove the lime odor. In the context T, so this is a context that is really similar to the context A, except that we remove the lime odor. Here in the context C, we dim the light, we remove the lime odor, and we remove the sound presentation. So this one is really different from the conditioning context, and we recreate the conditioning context at the end. So this is basically the behavioral protocol. So as again, the animals stay in the same place, and we just remove some contextual elements. And this is the behavioral result we got. So again, we measure the freezing behavior in those animals. And we have the A, B, C, A context. You see that they freeze to the conditioning context A. They generalize to the context B, but they discriminate to context C. And when you put them back in the A, they freeze again. So we have designed a behavioral paradigm that allows us to create a generalizing context and a discriminating context. And using this paradigm, we know that the mice generalize to context B, whether they discriminate to context C. We have done one control experiment for this behavioral paradigm. As you can see, because we expose sequentially the animal to A, B, C, A, you may say, okay, this effect is due to could be due to an extinction process because animals spend 9 minutes in the context, so it's not fearful anymore. So we perform the same experiment, but without touching the context. So the animals were tested for 12 minutes in the context A, and you don't get any extinction. So you cannot explain this effect here by an extinction. Okay? So contextual discrimination cannot be explained by extinction. We have done different behavioral controls as well in this paradigm. So again, we have day 1 habituation, day 2 conditioning, and the sequential exposure to ABCA in day 3. The first control we have done, we have exposed again the animal to the ABCA context on day 4. And this time we wanted to evaluate whether the low freezing level in context C was due to novelty effect. It could be explained simply by the fact that the animal is the first time the animal is exposed to the C context, so they could reduce their fear because it's a novel context. So you can see that on day 4, if you re-expose them to ABCA, we get back the same effect, so it's not due to novelty effect. And we also control if it was due to a timing effect, the time when we presented the context C. This is in the third position, but what about if we presented it in the first position? This was done on day 7 by exposing the animal to CA context, and we got the low freezing level each time you expose the animal in the C context. So those results cannot be explained by a novelty effect or a timing effect. Okay, this is the conclusion from it. And we have used, again, single unit recording from the dorsal part of the prefrontal cortex. Here you have an example of a lesion to identify the recording site in the prefrontal cortex. And again, we have separated the neuron into putative, excitatory, and inhibitory neurons using the same parameters we have used before. And we end up with the same proportion, roughly the same proportion of neurons corresponding to inhibitory interneurons or excitatory neurons. So again, you have the behavioral result here for the experimental group ABCA. So the gray here is fear generalization in context B, discrimination in context C, and fear again in context A. And you have the control group exposed to A only, where you don't have any effect. And at the same time, we perform the recordings in yeah.
Can you counterbalance the removal of the cues, like first the sound and then the odor?
No, we haven't done that. What we have done for the behavioral protocol is what I think that's an interesting observation. We observe that if you add contextual elements, the animal cannot discriminate the different contexts. They are able to discriminate when you remove sequentially the elements. So that's what one first experiment we have done. If you remove, let's say if you have a neutral context and you add the sound, the line odor, they won't discriminate at all. So this was important first to remove the elements one by one, but we haven't tried to start with the light or the CS or the line odor. We don't know that. So we have done recordings during the ABCA exposure and the AAAA exposure, and this is what we observed. So you have the normalized firing rate of the 121 neurons recorded in ABCA in blue, or 48 neurons recorded in AAAA. What we observed is that there was no change in the firing rate, no significant change for the control group. But for the experimental group, we observed that when we expose them in the B context where they're generalized, there is a small increase in the firing rate. But more interestingly, when we expose them to the C context where they discriminate, there is a huge increase in the firing rate of those neurons. Okay, and somehow it comes back to baseline when re-exposed to the A context. So this is an example of such a neuron. You have each yellow bar is the firing, is one single action potential from one neuron. During the A, B, C, A prime exposure, you see that in B context, the neuron start to fire and increase its firing rate mostly in the C context here. So this is a neuron that does exactly that. So the conclusion from that is that the contextual fear discrimination here is associated with an increased activity of putative prefrontal excitatory neuron. I didn't mention it, but we concentrated on the excitatory neuron for this experiment. This is a different way to look at the data in a more dynamic manner. We use principal component analysis to identify the firing patterns of the neurons recorded in the prefrontal cortex in the ABCA exposure. And you see that this is a subset of 30 neurons that were strongly activated during the C context. And you can see they are not activated in A, not really more in B, but strongly activated in C, and they go back to baseline in A. This is the quantification of it, where you see the strong increase here. And this is another way to represent the data. You have the 30 neurons here. Each line is a neuron, and in black or white you have the firing activity of the neuron. And you can see that there is a massive increase in the firing activity to the C context. Okay, so we wanted to control a few things about those neurons, whether or not the change in neuronal activity was due to novelty or timing effects. So here what we compare the firing rate of the neuron in the C context on the first exposure to the C context or the second exposure to the C context. There was no significant difference. And we also compare it with the third and the fourth exposure, and there is no difference in the firing rate of the neurons that are activated in the C context, indicating that the neuronal discrimination we see in context C is not due to novelty or timing effect. You could say also that, if I go back to the behavior 1 second, because the animal frees less in the C context, one could say that the increase in neuronal activity is simply due to the locomotor activity of the animals, right? That could be simply related to a motor effect and not to contextual discrimination. And to control for that, we look at the velocity of the speed of the animal during the no freezing period in A, B, C, A prime context. And you see that in the C context, there is no change in the velocity between the control group and the experimental group, indicating that the increase in the firing rate during discrimination is not due to a change in the speed activity of the animals during the non-freezing period. So this demonstrates that putative prefrontal neurons increase their firing rate during contextual fear discrimination.
Can I have one more question?
Go ahead.
Cannot leave a day and cognitive load, because first you ask the animal to remember one thing, but you remove just one characteristic cue, and you add another one. So now the animal has to update and has to, in a working memory, that there is two characteristics missing. So there is increasing.
So how would you control for that? I see your point, but I don't see how to control for that. That's the cognitive load. Yeah, I don't see how we could control for that. Well, basically, I think we need to, if we want to address this question, we need to have a control situation or where we remove two elements, and but without behavioral effect, right?
Or maybe if you would remove the third element, it would be, even more increase in cognitive load.
It could be that. Yeah, but you would predict the same result. Okay, let's say if you remove 1, 2, or 3 elements, if it is contextual discrimination, you expect exactly the same thing that if it is a cognitive load. So that's
to be a task different condition.
Yeah, or something we haven't tested at the moment is as you suggested before, is to remove the elements in a different order. Maybe it make a difference in terms of behavior. Maybe they discriminate less if you remove the sound first and
without adding, because you mentioned adding would also represent it now it's an update.
Yeah. So that's
but
the
less cognitive load because it doesn't have to require.
Well, the question then, can you compare the removal of 2 elements with the adding of 2 elements in terms of cognitive load? I'm not sure if it is equivalent, I would say. Although if we assume it is equivalent, then we have we have the data and we know that they do not discriminate. Okay, so this was the basic observation, and the next question we were interested in is where do these discriminating neurons project to, and can we specifically manipulate them? So again we use optogenetic manipulation and identification. So from the mPFC I mentioned, then the 2 main regions involved in conditioned fear behavior are the basolateral amygdala or the ventrolateral bag. For instance, if you manipulate the ventrolateral bag, if you perform electrical stimulation, you induce freezing behavior. So there was potentially two structures to target, and we wanted to manipulate first the mPFC to the VL-PAG pathway. So we use mice that express the Cre recombinase under the CAMK2 promoter, and we injected an AAV virus expressing the archaeorhodopsin or channelrhodopsin in the mPFC, and we implanted the optic fiber in the VL-PAG to manipulate the fibers. So this is an example of the labeled neurons in the cingulate and prelimbic area, and we have the optic fibers in the VL-PAG here, above the VL-PAG, and the labeling of the fibers in the lateral and VL-PAG. So before conditioning, what is the effect of the inhibition of this pathway? So before conditioning, we compare the pre-laser stimulation with the stimulation and the post-stimulation for the GFP animals and the archaeorhodopsin-infected animals, and we don't see any change in the locomotor activity. So there is no change in locomotor activity before conditioning. What about post-conditioning? So here you have the A, B, C, A prime. Open is the GFP. So the left bars are GFP, and this one is the arch T. So the right bar are the arch T. So we manipulated, of course, we are interested in the fear discrimination. We know the neurons from the mPFC are activated during discrimination. So this is the normal GFP control group. If you inhibit those if you inhibit those neurons, you induce a fear generalization. Again. So the optogenetic inhibition of DMPFC inputs to the VL-PAG induce fear generalization. We did the opposite manipulation. We activated the fibers from the mPFC to the VL-PAG, but this time in the B context when the animal generalized. If you activate those neurons when the animal generalizes, you induce a reduction of conditional fear responses, so you induce discrimination in those animals. So this demonstrates that the optogenetic activation of DMPFC inputs to the VL-PAG induce fear discrimination. Okay, so I will come now to an important question in terms of optogenetic. So this is the strategy that a lot of people have been using in the past, that you infect neurons and you manipulate the fibers in the target structure. There is one strong limitation of this approach, is that you may also activate fibers that are not ending in the structure, but that just pass by the structure and end up in a different structure. So this is a critical, it could be an alternative explanation of the result. You do not impact the VL-PAG, but you impact just en passant fibers. So it is important to control for that, for this kind of experiments. And I will describe the strategy we have been using for testing specifically this hypothesis. So instead of manipulating the fibers that project to the VL-PAG, we use the opposite strategy. We injected a retrograde virus in the structure that expresses the Cre recombinase. So the Cre recombinase is taken up by the axon and travels back to the soma. And in the soma, we injected the AAV virus that recombine with the Cre recombinase. So you infect specifically the neurons that project to the structure you are interested in. And then you put the optic fiber in in the prefrontal cortex in this case. And this is an example of back-labeled neurons in the cingulate cortex and the prelimbic area following an injection in the VL-PAG. So you can clearly identify neurons and manipulate those neurons. And this is a more specific approach.
Good question. When you do that, those neurons don't have collaterals to the BLA?
Yeah, but because you manipulate those neurons only in the...
No, I'm just saying if the neuron that goes to the PAG also goes to the BLA.
Ah, okay. So we do have a control for that. So what we have done, we use simply retrograde tracing. We injected retrograde tracer in the BLA and the BL-PAG simultaneously to see if they colocalize, and they don't. And I can add on this, we know that the BLA, the neurons in the mPFC that project to the BLA are localized in the superficial layers, and the one that project to the VLPAG in the deep layers. So they are really segregated. Sorry, what am I missing here? Okay. The one question here that was missing for us is whether or not the neurons that are activated during discrimination are the one that project to the VLPAG, right? So to address this question, we use this strategy I was just describing, and this is an example of neuron. So it is a neuron that projects to the VLPAG that we were able to optogenetically silence. So after when you remove the inhibition, you see the neurons start to fire again. And what was doing this guy? This guy was strongly activating the C context. So this is just an example we are following on that. But it suggests that the neurons that are activated in the C context during discrimination are the ones that project to the VLPAG. Of course, we have used the same strategy, but to try to manipulate the neurons that project to the BLA to see if it was two complementary pathways involved in the discrimination. So we injected the CAF3 virus in the BLA and manipulated the soma of the neurons in the prefrontal cortex. After injection of the AAV virus in the prefrontal cortex. And this is still preliminary, but we have manipulated either in the B or the C context. In gray, we manipulated the B. This has no effect. And when we manipulated the neurons in the C context, this has no effect either. So we didn't find any change in terms of freezing behavior upon the optogenetic inhibition of mPFC neurons that project to the BLA. So that was the story. So we're following a bit the experiment on the amygdala and increasing the N also, but this is not yet published. So to summarize this part of the presentation, we have developed a behavioral paradigm that allows single units recording during context sphere generalization and discrimination. The putative excitatory neuron displayed enhanced activity during contextual fear discrimination. If you activate the DMCFC VLPAG pathway, we promote fear discrimination. If we inhibit this pathway, we promote fear generalization. So we think we have identified a critical role of this pathway from the mPFC to the VLPAG in mediating behavioral transition between fear generalization and fear discrimination. And again, you have seen we have always used the same strategy. First, we'll record the neurons, we try to identify what they are doing, and we try to perform the optogenetic manipulation, matching exactly the changes in activity of the neurons. I have finished what I wanted to show you. I don't know what's time for questions. I just want to finish by thank the people in my lab that have been working in those projects and my funding agencies that helped me on that, and I thank you for your attention. So something I didn't mention, so I'm part of the same institute than Pierre Vincenzo Piazza in Bordeaux, the Neurocentre Magendie, and we got recently some French grants to develop a platform for behavioral neurophysiology. So we have set up a full range of behavioral tasks, and each of those tasks, it could be addiction, it could be fear conditioning, it could be water maze, whatever. We have associated it with single units recording and optogenetic manipulation. And the goal of this platform is also to provide formation to students. So in the coming year, we will probably propose some courses on behavioral neurophysiology and optogenetics. If some of you are interested in learning how to record neurons and identify and manipulate them simultaneously, then just contact me and I will give you the information. Yeah.
This is just a general question. So for the first part, you mentioned there was no optogenetics really at the time, so you didn't use it. And by, I think it was 2014, the next part that we showed, you've done some very nice work with it. How long did it take to get established and functional optogenetics working in your lab?
So my lab was created in 2011, but I've been trained already during my postdoc to optogenetics, so it was quite straightforward for my case to set up the so optogenetic alone is not so complex. I mean, it starts to be complex when you perform the simultaneous manipulation and identification, because you need to construct electrodes, to put electrodes around the optic fibers, and then it can get quite complex to record the neurons at the same time. But once the technique is established, once you have nice electrodes, nice optrodes, then you can, it's quite obvious. Yeah.
Yes. So I have a naive question about the theta oscillation.
Yeah.
So in the model, there's sort of it's like a binary switch, so asynchrony and then synchrony. Is there any does the degree of asynchrony have any effect, or is it really a binary, either it's synchronous or asynchronous?
So is it binary in the way I've shown it? Because I took two examples where animals were showing low fear or high fear. But of course, you need to think about that in terms of a continuum. Sometimes you have animals freezing only at 50%, sometimes 80%. And of course, you have a pure correlation between the freezing behavior and the degree of synchronicity of the neuron. So it's not binary as I discussed. Yeah.
So Barcelona and the projection to medial frontal cortex and especially to prelimbic area not only encodes aversive stimuli like associated to frequency, but also reinforcing stimuli. Do you plan to do the same work?
So that's a really good question. We had some plan at the beginning, but to study simultaneously appetitive and aversive conditioning. And the idea was to submit the animal sequentially to conditioning aversive task and appetitive task, and to record the same neurons to see if they were active in one or the other. That's a pretty tough experiment, you can imagine, because you need to follow the neurons for maybe 2 weeks, and this is not trivial. But I know in my previous lab, in Andreas Luthi lab, they have designed a task that is purely Pavlovian, where the animals are first fear conditioned, and then they are the next test. The next day is an appetitive conditioning in a Pavlovian manner, with an with a delivery of sucrose directly into the mouth, and the sucrose delivery is associated with the sound. And so they have followed the same neuron during this task, and the output is that you have a kind of segregated population. You have only a small proportion of the neurons that are activated in both tasks, maybe 5%. But it seems to
at the level of basal or amygdala.
Exactly, not the prefrontal cortex. This was in the basal amygdala, not in the prefrontal cortex. But it's the same. The basal amygdala has been involved in aversive and appetitive learning as well, via projection to the accumbens or to the VL5. So in the prefrontal cortex, we haven't done it. I know some people are working on that at the level of the amygdala. And the output of those studies so far is that you have segregated population activated either in fear conditioning or appetitive task, and only a small fraction of the neurons that overlap. Another interesting output of those studies is that the order of the conditioning, whether you start with the appetitive or the aversive, can profoundly impact the selection of the neurons. So the neurons that are, if you start with aversive conditioning, you bias the neurons to, you have a stronger proportion of the neurons that react to aversive conditioning rather than the appetitive one, and it is the opposite if you start with the appetitive. That's what I know about this kind of experiments. Yeah.
Just in answering his question, you had said it'd be very difficult to follow a neuron for 2 weeks. How long can you follow?
So again, you record the neuron extracellularly. So that's you cannot be really conclusive about that. The way we face this problem is that from day to day we compare the waveform of the action potential, the average waveform of the action potential, and we make correlation between the two from day 1 to day 2, let's say. And if we have a strong correlation coefficient, we consider that we are recording from the same neurons. So this works for a structure like the amygdala. So if you start with a pool of 100 neurons, after a couple of days you end up with 2,500. But those are really stable, and those guys you can keep them for maybe 3-4 weeks. Again, based on the correlation measure we have done. So usually after 2 days you go down to 25 to 30%, and but you keep those guys for a long time. And from my experiment in the BLA, the longer time we could keep the same neuron was about 2 months. But again, some people will say that we have no guarantee that it is the same neuron. That's just the limitation of the technique. Okay, so I think we should. If you don't have any more questions.
So we have to be here at 4:40 to have our next presentation. And we have